All right, let's jump right into this explainer. Today, we're decoding the ultimate blueprint of human identity and looking at exactly how our traits are passed down from one generation to the next.
I mean, think about it for a second. How is your eye color actually decided? Or your natural height, the texture of your hair, or even your susceptibility to certain genetic diseases? These aren't just random coin flips. They're the result of highly specific, real-time instructions. And to understand how they work, we really need to look microscopically close.
Well, the answer to all of that comes down to one tiny, absolutely awe-inspiring molecule, DNA, or deoxyribonucleic acid. You'll find this genetic material packed into the nucleus of almost every single cell in your body, with a little extra hanging out in your mitochondria. You can basically think of DNA as the ultimate instruction manual. It's the book of life that tells our bodies exactly which proteins to build and precisely when to build them.
All right, moving on to Section 1, the alphabet and chapters inside the double helix.
If you were to literally zoom way in on this molecule, you'd see it looks exactly like a twisted ladder, which is famously known as a double helix. This whole structure is made up of long polynucleotide strands, and you probably know it was modeled back in 1953 by James Watson and Francis Crick. But, and this is a huge but, it's absolutely crucial to mention that their breakthrough was heavily aided by Rosalind Franklin's brilliant X-ray diffraction images. Her work was literally the key that unlocked this visual for the entire world.
So what's actually making up the rungs and sides of that ladder? Well, if you break it down, every single nucleotide, which is the most basic unit of DNA, is made of just 3 ingredients. You've got a deoxyribose sugar, a phosphate group, and a nitrogenous base. These 3 components link together in repeating patterns to form the literal building blocks of our genetic alphabet.
And this brings us to the complementary base pairing rule, which is honestly kind of like a molecular lock and key. Out of those nitrogenous bases, we only have 4 letters in our genetic alphabet: adenine, thymine, guanine, and cytosine. But they follow really strict rules to keep that twisted ladder intact. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. It's a perfectly complementary system that securely holds the 2 sides together.
Section 2. Packaging the code. Genes and chromosomes.
So, zooming out just a bit from those individual letters, we see that these sequences actually form functional segments called genes. And one single gene holds the information for a specific protein or specific trait. But you can't just have loose DNA floating around, right? It has to be organized. So it wraps really tightly around special proteins, called histones, to form dense packages known as chromosomes. To put that into perspective, humans have exactly 46 chromosomes in total, neatly organized into 23 pairs. 22 of those pairs are autosomes governing most of our body's traits, and that final pair, those are the sex chromosomes that determine biological gender.
Next up, Section 3. Reading the manual. The central dogma.
Okay, so how does your body actually read this manual to function? We call this the central dogma of molecular biology. Basically, it's the master process of moving from DNA to RNA to protein, and it happens in 2 main steps. First is transcription. Your DNA is way too precious to leave the nucleus, so its information is safely copied into a temporary messenger called mRNA. Then comes translation. That mRNA travels out to a ribosome, which reads the message and starts linking amino acids together, kind of like beads on a string, to build a protein. And proteins are the real heavy lifters here. They control almost every single biological activity inside you.
Section 4. Copying the code. DNA replication.
Now, what happens when a cell needs to divide? Obviously, exact copies of the manual have to be made. The cell uses an incredibly specialized crew of enzymes to do this. First, an enzyme called helicase swoops in and acts like a zipper, opening up the double helix. Next, DNA polymerase comes in and builds the brand new complementary strand based on those strict pairing rules we talked about earlier. Finally, ligase acts like glue to smoothly join any fragmented pieces together. We call this semi-conservative replication because every new DNA molecule keeps one old original strand and gets one brand new one. The mechanics of it are just remarkably efficient.
Section 5. Typos in the code, mutations.
But hey, nobody's perfect, right? Sometimes the DNA sequence undergoes a spontaneous change during that copying process. We call this a mutation. Now, the word mutation usually sounds super intimidating, mostly thanks to sci-fi movies, but it's actually the very engine of evolution. Mutations generate new genetic variations. Sometimes they're completely neutral, with zero effect. Sometimes they're actually beneficial and help an organism adapt to its environment. But of course, they can be harmful too. Serious genetic disorders like sickle cell anemia, hemophilia, or cystic fibrosis are all the direct result of mutations, literally just a tiny typo in the code that alters how a crucial protein is built.
Moving on to Section 6, Beyond the Body, DNA in the Modern World. Stepping back to the macro level, this knowledge has leaped way out of the biology lab. It's a real game changer out in the real world. At a crime scene, DNA fingerprinting is the absolute gold standard. In a hospital, we're predicting diseases long before they even manifest. From genetic engineering and rapid vaccine development to tracing the literal evolution of biodiversity across the globe, DNA technology is basically the backbone of modern science.
Finally, Section 7, Mind-Bending Biological Scale.
To wrap up this explainer, brace yourself for some staggering facts about the microscopic universe living inside you right now. If you actually sat down and counted the letters in your personal instruction manual, you'd find approximately 3.2 billion base pairs contained within the human genome. 3.2 billion combinations just to make you, you.
But here's the real kicker. Despite that unimaginably massive number, 99.9% of that DNA is completely identical across all human beings on the planet. That tiny, minuscule 0.1% difference is what accounts for every single variation in eye color, height, and physical appearance among the billions of people on Earth. We are literally so much more alike than we are different.
And physically speaking, this is wild. If you took the tightly coiled DNA from just one single microscopic cell in your body and stretched it out perfectly straight, it would be about 2 meters long. That means if you took all the DNA in your entire body and stretched it end to end, it would stretch for billions of kilometers. That's far enough to reach the outer planets of our solar system and back. No joke.
So, what does this all mean for us? Ultimately, DNA is the blueprint of our biological identity. It is life's original language. Understanding this magnificent code isn't just about biology, it's about decoding the very essence of life itself. The question is, now that we know how to read the ultimate instruction manual, what are we going to do with it? That might just be the greatest question of our modern age. Thanks so much for joining me on this explainer, and keep exploring.